everybody, we're going to go over transcription and translation, which you've been starting to look at through your HyperDoc, the Edpuzzles, and all those other videos. So we already are masters at DNA replication. We're going to talk about how we go from DNA now all the way to a protein, and that's through these processes, transcription and translation. So before we start anything, we have to talk about the types of RNA that we're going to see. So there's messenger RNA, which is also called mRNA. It carries a copy of your instructions from that DNA to the rest of your cell. Ribosomal DNA, that's also called rRNA. It makes up the two subunits of ribosomes, and that's where proteins are actually assembled. And then there's your transfer RNA, which is also called tRNA, and that has the ability to transfer amino acids from the cytoplasm to the ribosomes so that it can make this long amino acid chain, which is your protein. All right, so the first thing that needs to happen is transcription. That's your RNA synthesis. So we make mRNA during transcription, and we use DNA as the template. Your new RNA strand complements that DNA template. So we're going to try that out below. You're going to use this template DNA sequence that I have written for you here to try to transcribe and form mRNA underneath. Remember, there are a couple of important differences between DNA and RNA. And one of the most important ones here is that DNA has thymine, T, but RNA has uracil, U. So anywhere that a T would have matched up, we're instead going to put a U because we're making an mRNA sequence. Let's do the first one together. The first three letters are TAC. T binds with A, so your first letter of mRNA will be an A. A normally binds with T, but we're working with RNA, so we're going to replace that with a U. So you have A, U, and then C binds with G. So in this first box right here, you should have A, U, G. I want you to pause the video and try to fill in the next few boxes on your own and replay the video once you have it. Hopefully this is what you got for your answers. We're just matching up our base pairs, but anytime that you would write a T down, instead you're writing a U for uracil. All right, transcription happens in the cytoplasm in prokaryotes, and in eukaryotes it happens in the nucleus, and then your new mRNA chain moves into the cytoplasm for the rest of protein synthesis. There are three important steps. First, your RNA polymerase will bind to the DNA and separate the strands. Then a promoter sequence tells the RNA polymerase where to actually start working. Then that RNA polymerase actually starts using the DNA template to build a new nucleotide chain of RNA. All right, once transcription is over, we move to translation. But before we talk about that, we have to look at this thing that we call the genetic code. It's how we read mRNA. So we read it 3 letters at a time. Those 3 letters are called codons. So each codon, or 3-letter like little word, codes for 1 amino acid. And there are 20 total types of amino acids. And the way that you link them together, the order that they're in, the type of amino acids that you put together, that builds up one polypeptide chain, which is also known as a protein. All right, so you have these 3-letter words, these 3-letter codons, and we have to be able to figure out what each codon actually means. So we use this thing called a codon wheel. It's read from the center out. So your first letter has to be one of these 3 in the center, and then your second letter, you move into the next box of whichever section you're in, the next level down. And then the third letter, you go 1 level down below that. You just keep working your way out. So let's try it for the amino acid codon. Let's try to find the amino acid for the codon AUG. We're going to start with A in the center, then move out to this U that's in the A section. And then last, we go to this G right here. So if you do that, AUG codes for methionine. That's the amino acid that it codes for, and that's the start codon. It tells translation where to start. So what I want you to do is pause this and try to find the amino acid for the codon UGG. All right, if you start at U right here and go GG, you should find that the amino acid it codes for is tryptophan. All right, translation is when our mRNA is decoded to make a protein. So it's protein synthesis. It happens in the cytoplasm. Your mRNA is transcribed in the nucleus, and then it moves to the cytoplasm for translation to happen. Your translation has to attach, your mRNA has to attach to a ribosome for translation to actually begin. So these are the steps that we need to know. First, your ribosome attaches to the mRNA. Then it reads the mRNA and directs tRNA to bring the correct amino acid into the ribosome. And your ribosome can hold 3 amino acids at a time. You can kind of see that in this video right here. So those tRNAs keep bringing in amino acids and making and building this long polypeptide chain, which makes a protein. Your process stops when the ribosome reads a stop codon. So when you look right here in this picture, UGA is a stop codon, and that allows the ribosome to release the polypeptide chain so it can fold together and make a protein. All right, hopefully this helped clear up any confusions you have. I hope that you have a great day.